Welcome everybody to a Striatech hosted journal club. My name is Thomas Münch and I'm one of the founders of Striatech. I welcome you to another of our journal clubs where scientists and researchers present their projects and data, highlighting the use and application of our products. Striatech is a dynamic biotech company from Tübingen, Germany. We specialize in neuroscience testing tools, in particular for vision research. The Striatech founders are all experienced neuroscientists in the field of vision research. Today, we welcome Anna Matynia as our speaker. Her talk is entitled Visual Function with Aging in Normal and Alzheimer's Disease Model Mice. Anna has just recently been appointed Associate Professor at the Department of Vision Sciences, College of Optometry, at the University of Houston, where her work is focused on neural circuits that are associated with clinically relevant diseases of both retina and cornea. She can look back on diverse research experience from her Ph.D. work on the cell cycle in yeast at Baylor College of Medicine in Houston, to the studies on behavioral aspects and the molecular underpinnings of learning and memory in mice at UCLA. Staying in Los Angeles, she switched her focus to research on the neurobiology of the eye, focusing on clinically relevant questions. This included light-induced pain, damage to the corneal surface, or changes to the retina during neurodegenerative diseases such as Alzheimer's. The latter belongs to the line of research that we'll be hearing about today. Before you start, Anna, let me remind the audience that you will be able to ask questions throughout the event and also give a thumbs up to other questions. We will get to those questions at the end of Anna's presentation. When you want to ask a question, the cursor by default is in the name field. Giving your name is optional, and you have to click into the question field before you start typing your question. The Q&A panel on the right will stay available for a couple of days after the live event, so if you are currently not watching live, but a recording of the event and Q&A is still available, please make sure to enter your question and your name so that we can get back to you with the answers. We are very much looking forward to your question and a lively discussion at the end. So Anna, please go ahead and turn on your camera, unmute your microphone, and share your presentation with us. And I'm looking forward to hear about your work.
Good morning and thank you. Let me just put this in presentation mode. There we go. Okay, so thank you very much for allowing me to present here on the work that we've been doing over the last couple of years in visual function in normal aging in Alzheimer's mice. This is the first time that I will have talked about this in public, so thank you for your patience in that. You'll see that some of the result details are ongoing experiments. Okay, so it has often been said that our eyes are the windows to our soul, but one thing that is definite is that they are a window to our health. And this starts from the earliest moment in childhood with a newborn baby where you can look for a red reflex, which is basically being able to see through the cornean lens to the back of the retina. And this can be used to diagnose potential blinding diseases in nonverbal infants. But after we're born, the only thing that happens to us is that we start to age. And with aging, fortunately, typically decades later, we start to have other pathologies that affect our vision. And these are, I mean, I call them pathologies, but they're just normal aging, so non-disease associated visual function changes. Let me just give myself a pointer here. So, for example, as we get older, our lens starts to get denser and undergo discoloration. When this becomes a problem, we end up getting cataract surgery as it reduces light transmission and affects our vision. The retina, as we age, and typically, again, into multiple decades in, we start to see disorganization of the neurons in the retina and neuronal loss. This can affect visual acuity, the pupillary light reflex, which is when the pupil constricts when you shine a light on it, and multiple other functions. And then, of course, in the brain, the parts of our brain that process visual signals also undergoes neuronal loss. And this can affect many, many functions, as can the retina: contrast sensitivity, binocular summation, motion sensitivity, and the list goes on. So this happens normally with aging. Most of us start to notice this when we need reading glasses. So in our 40s or 50s, when we start to experience presbyopia. But a lot of times you start to see these deficits maybe in the 7th or 8th decade. So that's good news for us. However, the thing that can happen is you can have a disease that will hasten this visual decline. And this can affect specific functions depending on what areas are affected or what cell types are affected. So it's really important to think about the retina as a window to our brain. It is part of the CNS, and that we can use retinal biomarkers to provide early detection capabilities, since it is optically accessible non-invasively. So that's a really big advantage of looking at the retina, is that we have all these tools for both imaging and functional evaluation. And this can perhaps help us with the evaluation for onset, control, and progression of disease, whether it's a disease that we know how to treat or one that we are still searching for solutions. And of course, the basis of this is we need to have a good comprehensive analysis of visual function during normal aging, against which we can then compare the disease state. So for those of you that, I apologize if this is pretty basic, but let's just go over the photoreceptors of the eye so that we can start thinking about where these visual disturbances can occur. So we have your classic photoreceptors, the rod photoreceptors shown in blue, that are monochromatic. They do low illumination, or your what we would typically think of as night vision. We have cone photoreceptors, and these are shown in red. These are our high acuity daytime color vision. And together these both detect photons that signal through the inner retina and then end up transmitting their information to retinal ganglion cells, which are the outermost neuron of the retina before it goes projects to the brain. So there are two major groupings of retinal ganglion cells in the schema that I'm presenting here. One is part of the neural circuitry for transmitting information to the brain, and the other one contains melanopsin, which is able to detect photons intrinsically. And there are many light-dependent functions that are independent of classic vision, so are dependent on these ipRGCs. So I'll call them ipRGCs for intrinsically photosensitive retinal ganglion cells. These were first identified back in about 1990, and mouse models were fast to come. They were identified in the 90s, and then mouse models started to be developed and published in the early 2000s. So so already you can see that we have three distinct pathways for light transmission and the type of information that we might use to function. These ipRGCs and RGCs project to the brain via the optic nerve. And for the most part, most of the retinal ganglion cells project to the lateral geniculate nucleus that then projects to higher visual functions in the visual cortex. However, ipRGCs are much more diverse in their projections, and there are 5 or 6, depending on your classification scheme, different types of ipRGCs called M1 through M6. And as you can see from this summation, these project to multiple different regions that underlie different specific functions. For example, the M1s, most of the M1s project to the suprachiasmatic nucleus that underlies circadian photoentrainment. A number of the classes project to parts of the geniculate nucleus, and then some of the M1s also project to the olivary pretectal nucleus, the shell or the core, and this is responsible for the pupillary light reflex. So you can see that there's a diversity of functions that might be affected depending on whether you have a a condition that affects rods, cones, ipRGCs, or the processing centers that they project to. Okay, so let's just talk a little bit about these retinal ganglion cells and specifically the ipRGCs. So it's been shown, the references are down in the corner. It's been shown that in normal aging, we see a loss of ipRGCs. And this is just a small image of human retina under 30, 30 to 50, 50 to 70, and over 70 years old. And you can see that you start to see loss of these ipRGCs around in the 5th decade of life. But one of the things that had been known about ipRGCs was that they were generally resistant to trauma. So in an optic nerve crush, other regular retinal ganglion cells, RGCs, would die from optic nerve trauma, but ipRGCs tended to survive. So it was thought that perhaps they were resistant to disease and injury. However, it was identified by Chiara Lamarja in human Alzheimer's disease retinas that ipRGCs were actually more sensitive to cell death in Alzheimer's disease. And this was borne out in both human and mouse models. So I'm showing you here an image of a human post-mortem retina, age-matched with an ipRG soma and dendrites shown, and then an Alzheimer's patient where you see a loss of a sickly cell that may be in the process of dying and decreased melanopsin function. Now, my colleague Luis Perez de Sevilla-Muller showed that in a mouse model of Alzheimer's disease, and this is a double transgenic model that you could see in healthy controls, nice dendritic trees of the melanopsin cells in a 10-month-old animal. But if you look at the AD mouse model, you see a loss of this. So this early degeneration of ipRGCs is consistent from humans to mouse models. And from our perspective, we're very much interested in looking at symptoms and conditions that are mimicked in both human and mouse so that we can get greater insights into the human disease. So the question we had is, can we leverage ipRGC loss for early detection to provide non-invasive tools for clinical evaluation of Alzheimer's disease? As we know, traditionally Alzheimer's disease is diagnosed pretty late when you already have cognitive impairment, but that a lot of the pathology that leads to this has been set and been at work behind the scenes for about a decade. So the earlier we can detect it, the more likely it is that we're able to provide some sort of treatment for these patients. Okay, so today I'm going to talk about assessing outcomes in wild-type mice and in AD mouse models. For the most part, I'm going to show you data on this 3xTG Alzheimer's mouse model. It has 3 humanized mutations in it. One is of presenilin-1, which is the enzyme that cleaves amyloid precursor protein. This is your protein that makes the A-beta plaques that are known to be pathological in deposits in brains of Alzheimer's patients. And then it also has a phospho-tau mimic, and phospho-tau is also found in the plaques and neurofibrillary tangles in deposits in brains. So we use this mouse model because it has a relatively normal lifespan. We've done a little bit of studies. I'm just going to click back here to this double transgenic mutant, but it tends to die really early and it's difficult to have enough animals in it. So we've done a little bit of studies in these double transgenic mice, but most of the data is from these 3x transgenic. The only difference between these 2 mouse lines is the 3xTG has this tau, phospho-tau mutant, whereas the double transgenic only has the presenilin and APP variants. So we know that this 3xTG has a relatively normal lifespan, which makes that easier for us. We know that it starts to develop plaques and tangles with A-beta deposits around 6 months, and there are learning memory deficits by about 6.5 months. So we assess the visual structure and function from 6 to 12-month-old in both mutant and wild-type controls. So what visual functions did we assess and why? So it's known that AD patients experience multiple visual dysfunctions. These include deficit in spatial contrast sensitivity, motion perception, color discrimination, visual loss, impaired ocular motility, reduction in the pupillary light reflex, and circadian alterations in melatonin, to name a few. The ones that I've highlighted in red are ones that have contributions from the ipRGCs. And this is not an exhaustive list by any means, but these are ones that are important to our talk today. So this means that we should be looking at biomarkers that evaluate rod, cone, and ipRGC structure and function. And we can do this using in vivo assays. We can do electroretinography, which is like an EEG, but for the eye, and optical coherence tomography to look at the structure, and this is like a CAT scan of the retina. We can also look at ipRGC dependent and independent behaviors that include light aversion, the pupillary light reflex, visual acuity, and contrast sensitivity. And finally, we can look ex vivo so that we can correlate not just the extent of loss, but the timing of retinal neuronal loss and ipRGC survival. The first thing that we did, and this is published work, was to establish metrics in non-pathological animals. And so let's get on to our first set of tests, in vivo ERG and OCT. So an electroretinogram is, as I mentioned, it's like an EEG or an EKG. We're measuring the electrical output at a whole tissue level, but we're doing it on the eye. So here you see an anesthetized mouse, and it's put in what's called a Ganzfeld dome. So it's dark adapted, but it's put in this dome, and we can flash bright light we can flash a series of illuminations to get the kinetics of response. And we put an electrode, we put a ground electrode on the mouse, and then we put a small electrode on the eye. So we're measuring whole globe electrophysiological ion flow. So this is an example of a trace that you would see with microvolts on the y-axis and time on the x. You can see we get a baseline. There's a little stimulus artifact where we flash, and then you can see this first deflection. This is called the A wave. And this represents the photoreceptor contribution. If we do this in dark adapted animals, it reflects the rod with dim light, the rod photoreceptor response. And then you see recovery of it, you see these oscillating potentials, and then we also measure this B wave. This B wave is now, as we go down the circuit, it's the responses from the inner neurons, from the inner retina. And so we can measure these two things very reliably, and we've looked at this in wild-type mice from 6 to 12 months old, and we can look at scotopic, means dark adapted, so we're looking at the rod photoreceptor responses. So the rod photoreceptor responses are very stable in wild-type mice over 6 to 12 months. And then when we look at the inner retina responses with the B wave of the rod pathway, we also see that this is very stable from 6 to 12 months. So when we look at the Alzheimer's 3xTG model, we've really only looked at 10 and 12 months. We can see that they're pretty stable between the 10 and 12 months, and this value is pretty similar to the wild-type animals. These are data that we're still continuing to analyze, so we'll have a slightly better analysis, but it looks like there's not a major ERG or electrical change in the retinal function. And this is not surprising at all, because when you look at patients that have photoreceptor loss, it takes quite a bit of photoreceptor loss before you see deficits in the ERG. So if we have mild degeneration, we might not see this in such a global metric. So the next thing that we did was to look at OCT. So as I mentioned, this is like a CAT scan. We put the anesthetized mouse in this holder, and we can move this lens right up against the eye. And then we can look at light transmittance through the eye to the back of the retina. And here's an image of a retina where you can see the back end of it at the bottom. This is the choroid, or the outside of the globe. And this inner line is the it's the centralmost layer of the retina. And this is where the retinal ganglion cells live. The photoreceptors are down here. This can be automatically analyzed into all these different layers of the retina, which I'm showing to you here. We get total retinal thickness, and we can break it down into all of these different smaller regions. And you can see that over blue to red, 6 to 12 months old in wild-type mice, the retinal structure is pretty stable. And again, this is only from 6 to 12-month-old mice, so this is not an old mice. It's a maturing older mouse, but it's not old age, and this is consistent with what we would expect in humans as well. So the analysis of the Alzheimer's 3xTG model is ongoing, so I don't have that to show you here. But grossly, I don't think that we're seeing major changes. And again, this is not surprising because this entire thickness of the retina is 200 microns, 210 microns or so. If we were to see major differences, we'd have to see at least a 20 micron loss for this technique to be able to detect it reliably. So that's a pretty significant shrinking of the retina. That's why we do the histological studies later on. Okay, so now that we've looked at the in vivo metrics, we're going to move on to behavioral metrics. So again, this is something where we perhaps have more sensitivity and precision in looking at specific pathways instead of looking at the retina globally. So we're going to look at light aversion, and I'm going to describe each of these tasks and show you the evidence that ipRGCs are involved with it, where they are. So light aversion is something that I've been working with for a very long time. This is the chamber that we use. There's two different chambers. Each has a light or open area and a dark area. And then a mouse can move freely between the two. We have an overhead light that with automated tracking. So we perform extensive habituation to effectively eliminate anxiety from this task. And we like this because it's a very simple innate reflexive response that is designed to test when light is aversive, as we can change this level from 0 to 2000 lux. So here, just let me take my marker off. I'm going to show you what this looks like at 0 lux. This is this is a video that's sped up. It is not mice on speed, but you can see under 0 lux, the mice can go back and forth freely. They spend a fair amount of time on the open side, and under 0 lux, they still come out, under 1000 lux, they still come out into the light side. Now, with our overhead tracking, we can measure this. This is just a frequency distribution, showing the percent time in light with 50% marked in the dashed line. So you can see that under 0 lux or no light, the animals actually prefer to spend more time in the open or 2B lit side. We can then test these mice under 2000 lux. And just for reference, a standard office will be between 200 to 400 lux. So this is very, very bright light. And you can see there's a slight leftward shift, but really not much of one. And what this tells us is that the pupil constricts and restricts light to our retina very effectively and to the retina of a mouse. When we then dilate the mice with atropine and test them under 2000 lux, so they can no longer constrict their pupil and they're getting full retinal irradiance, you can see that it shifts even more to the left. But even with this, mice are still spending some 20-ish% of their 25% of their time on the open lit side. So it's very difficult to just look at, say, the 2000 lux measure and say whether or not that mouse was light aversive, because a mouse that was spending 60% of the time under 0 lux, under 2000 lux without pupil dilation, maybe 50% of the time, and with atropine dilation at 2000 lux, maybe 45% of the time in the light. That mouse has a little bit of light aversion, but not a lot. If you think about the mouse that spends 60 seconds at 0, maybe 40 seconds with 2000 lux, and then maybe 10 seconds or less with atropine dilation and 2000 lux. So to make this more a measure of the aversion, we're looking at the differential. So we look at the time in light that they spend and we normalize it to the 0 lux. So we take the time in light at 0 lux minus the time in light at whatever test lux we use and divide by the 0 lux. So if they spend equal time, the same amount of time in the light at, say, 2000 lux as at 0 lux, this becomes a 0. So 0 is no light aversion. And if they spend no time in the light at the test lux, complete aversion is represented by a 1. So now we can just see wild type mice with, and the colored bars are with no dilation and white is with atropine dilation. Wild type mice increase light aversion with dilation, as do mice that lack rod and cone photoreceptors. But when you take mice that lack melanopsin cells, you see that they have no light aversion. So this tells us that these ipRGCs mediate innate light aversion. Okay. So what happens in mice as they age? So we looked at wild type mice at 6 and 12 months old, and these are C57 black 6 mice. If you want to get the details, it's at this publication that just came out in January. So we can look at the response with no atropine, and we see that increase with over the 6-month aging period, and then also with atropine. So they retain their increased light sensitivity with dilation, but it does go up a little bit without dilation over age. There's no effect of sex. And then if we look at mice that lack these ipRGCs, they essentially have no light aversion at 6 months old, with or without atropine dilation, and essentially no light aversion at 12 months old with atropine dilation, but a little bit of an increase in light aversion at 12 months old without dilation. So since these mice don't have ipRGCs, this increased light aversion has to be a downstream processing or dependent on rod and cone photoreceptors. So we felt that this differential in light aversion was an excellent indicator of whether or not ipRGCs were functional. So what happens in our Alzheimer's model? So we look again at the 3xTG, and this time we're using wild type mice. It's in a different genetic background. So if you look at so in black are the no dilation, and in white is atropine dilation. And the wild type mice, these mice are very much similar to the mice that I just showed you. At 1000 lux, there's no light aversion without dilation, but with dilation, you see an increase in light aversion. The case is not the same for the Alzheimer's disease model. They're more similar at these 2 stages. So this indicates that already by 6 months old, these Alzheimer's disease mice have a disruption of their light aversion neural pathways. So when we then look at 12 months old, we see these wild type mice actually lose their light aversion differential between atropine and non-dilated mice, but our mutants become much more light aversive. So there seems to be a hyperactivation of the ipRGC or downstream pathways. So this is, to us, indicative that there's a pathology that's already there by 6 months old. So this might actually work as a really good early marker for disease status, but that there continues to be, through the degeneration and the biological changes that are occurring over the next 6 months, there continues to be changes in these pathways. So understanding how these change actually would be really important for understanding the states of neurodegeneration and retinal degeneration. Okay, so the next thing that we looked at was the pupillary light reflex. And this is, as I mentioned, it's just what happens when we go from a dark adapted pupil where it's fully dilated, and this happens in both mice and in humans, to a low light and a higher light where you get constriction of the pupil. And here you can see in the top, these are wild type mice with low light and high light. You can see this medium to a pinpoint constriction. But when you get rid of the ipRGCs, you see a significant alteration in these constriction parameters. So we looked again at wild type mice at 6 and 12 months old, and we looked at wild type mice are in black, and you can see their pupil constriction stays very tight and controlled. This was a semi-quantitative analysis where 1 is equivalent to using the highlight, where you get very pinpoint pupils, and a 3 was equivalent to dark adapted, so fully dilated. So 2 is sort of less constricted than this. So we see with mice that lack ipRGCs, we see that this also is different from the wild type mice and is pretty stable over 12 months. And in human literature, the PLR is actually even if you have changes in pupil dynamics, your PLR is generally conserved up till the 7th or so decade. And then there's also no sex effect in here. So one of the things that was important, so that was a semi-quantitative. What we've moved on to doing is instead of just looking at maximum constriction, right? So whether it's pinpoint or fully dilated or partial, with the light held onto it, the dynamics of pupil constriction are much more complex than that. And this is just a diagram of what happens during pupil constriction. So there's rod, cone, and ipRGC contributions to it. So you start with a baseline pupil diameter, and then you turn a light on, right? So we get a light stimulus. The first thing we get is this very rapid constriction, and this is rod dependent. And then once we get down to a maximum amplitude of constriction, you see that this is actually not completely stable. It does slightly escape, and also it has micro constrictions in there. So this is held constricted through ipRGCs. So if you were to get rid of rod and cone photoreceptors, you'd see the latency increase and the slope would get shallower, and you might not get quite as large an amplitude of constriction. And then when you turn the lights off, the electrophysiological properties of ipRGCs, they continue to fire even after the lights go off. So you have this slow redilation phase, and there's this early post-illumination pupillary response, or PIPR, where the ipRGCs stop firing, but they still retain some level of constriction. And then this late where they retain constriction, and eventually over time, this will go back to baseline. And these two PIPR phases are dependent on ipRGCs. So this is a theoretical trace from a human, but we can do the same thing in mice. So we can semi-restrained mice, we can track their eyes, measure their pupil size. We can see a baseline. This is just left, right, and this is just example data. It's not even, it might be from an experiment. It's just an example to show you how we can do this. We can get a baseline, we can see the rapid constriction, we can see the maximum amplitude, and we can see an early response and then a later response of returning to baseline. And we can quantitate this. So with this fully quantitative measurement, we can look at all of these different phases of the pupillary response. We can look at rod photoreceptor and ipRGC contributions. So we're just starting the analysis with the Alzheimer's disease mice. So then that brings us to our last behavioral test that we did, visual acuity and contrast sensitivity. And since all of you know about Striatech, you'll recognize that I pilfered these off of their website, and we used their Striatech Optodrum to do this. So it's well known that in people, the contrast sensitivity function decreases with age. And this is just where you plot the contrast sensitivity, and this is just looking at the difference between black, white to gray shades. Versus spatial frequency, which can also be called visual acuity. This is, for us, we get the test of reading the letters from big to small. For a mouse, this is done by changing the thickness of the stripes from thick, contrasting black and white stripes to very thin stripes. So with age in people, this declines, and it does so in animals as well. So this is a theoretical contrast sensitivity curve. There are a couple of factors in here that are, and it fits a double exponential line equation. And there are a couple of features in here that will define an animal's visual performance. The cutoff here is the equivalent of the visual acuity, or what we would test in the mice in the Optodrum at 99.72% contrast, and we can do the staircase to see where its visual acuity comes out. And then we have this peak contrast sensitivity. So this is the inversion point of the curve, and this is another metric where the contrast sensitivity and the spatial frequency will define this curve. Okay, so starting with visual acuity. It's known that melanopsin and ipRGCs contribute to contrast sensitivity, but not visual acuity. So this is a couple of data out of Schroeder and Schmidt, 2 different papers, where they look at the acuity, and this is cycles per degree. I'm assuming everybody's a little bit familiar with it, but spatial frequency measures cycles per degree, and contrast sensitivity is usually a log. So if you look at wild type mice, they peak out in this experiment, these mice at about 0.4 cycles per degree. If you look at mice that lack melanopsin, the OPN4 knockout mice, you see that there's no difference in visual acuity. But if you look at mice where the transducin for rods or for cones are silenced, you see a loss of visual acuity. And this was also performed in the Schmidt paper. These are controls, the first 2 columns showing normal control mice. If you knock out melanopsin, there's not really a difference in visual acuity. And if you not just knock out melanopsin, but if you use genetic mechanisms to get rid of the ipRGCs, so this is the same melanopsin ablation mice that I've been showing you, and another group where a subset of them are ablated, you can see that there's no change in visual acuity. So this story is a little bit different though when you look at contrast sensitivity. Here's from the same 2 papers again, looking at visual acuity. You see that inverted curve that we talked about. You can see that the 2 control mice in the dark circles are have normal contrast, are the normal contrast sensitivity curve. And then if you look at mice that have melanopsin knockout or melanopsin cell ablation, you see a loss of visual acuity. And this is noticeable, but maybe similar to mice that lack rod photoreceptor function, but not quite as severe as mice that lack cone photoreceptor function. So the way people typically plot this is exactly as you see, and they'll do a repeated measures ANOVA, but then they'll do t-tests on single points, right, to show that they're different from each other. So we've looked at wild type mice, and these are looking at visual acuity at 6 months and 12 months. You can see the number of mice that we tested, and there's no loss in wild type mice with no effect of sex in wild type mice for visual acuity. And similarly, there may be a bit of a shift for enhanced function at 12 months, but this is probably just normal variability. And if you look at it, the peak occurs at about the same point. There's no effect of sex, but there's just fewer animals, so you see a lot more variability in there. So in our 3XTG mice, we've looked at 6 through 6 and 12 months old. These are wild type mice of the different genetic background. And again, there's no loss over 6 to 12 months old. And in the 3XTG mice, we also think that there's no change in visual acuity over the 6 to 12 months old. It looked like there might be a little bit of a drop happening, which is why we added a lot more animals to these later ages. So then our next task is to look at the contrast sensitivity, because this is the feature that is prominent in Alzheimer's disease patients. So as I mentioned earlier, there's this contrast sensitivity function where we're already looked at visual acuity, but we need to look at peak contrast sensitivity as well. But most people just look at a point along the curve and do a t-test, whereas we should really be fitting this to a curve and using those critical metrics, the cutoff, which is the visual acuity, and then looking at peak spatial frequency and maximum sensitivity. So basically getting the coordinates of the peak where the inversion occurs. And so we're just working on analyzing those data. You can see with 3XTG at 12 months old compared to wild type, they have slightly better acuity here. And then we have to take a look and see if we analyze all of the animals together, if we're seeing an effect of contrast sensitivity. So that's ongoing. Hopefully get that done in the next couple of months. So to just summarize the in vivo effects, there are no major alterations in wild-type mice from 6 to 12 months old with structural OCT or functional ERG. And these are very global measures, so you would have to see a lot of loss to see significant outcomes on these. We do, however, see alterations in light aversion that suggest a rod cone age-related change. And this is seen through increased light aversion in wild-type mice without pupil dilation, and also increased light aversion in mice lacking ipRGCs without pupil dilation. So this is perhaps a function of normal aging. And in fact, it wasn't until we looked at this across age, because we've been running these types of experiments for over a decade, and we collect our initial studies are of habituation are all the same. So we have these type of data for hundreds of mice, for the wild type mice at least. And we really didn't, from day to day, month to month, year to year, you don't pay attention to the age. So when we went back and retrospectively mined all of our data out, we were a little bit surprised to see that there is this change with age. So in the 3xTG Alzheimer's disease mouse model, we see abnormal light aversion by 6 months old. And we see continued changes in light aversion suggest that there's ongoing degeneration and alterations to the pathways. So far, we see no major alterations in the pupillary light reflex or visual acuity, and we're not sure about contrast sensitivity, but these are also still being analyzed and we'll know soon. So back to our overall goal, we've been looking at the in vivo measures. So then we want to correlate these to ex vivo retinal neuronal loss and ipRGC survival. So as mentioned, the in vivo structural analysis requires quite a lot of degeneration before it can show up. So to remind you, we do see a loss of ipRGCs with aging in the 5th decade and beyond, but that this loss is exacerbated in Alzheimer's disease in both humans and mouse models. So we looked first at the M1 class of ipRGCs at 6 and 12 months old, and these cells are located in 2 different layers in the retina. Most of them are in that ganglion cell layer, which is shown here, but some of them are displaced, and there are fewer that are displaced. So we looked at both types in males and females, and we saw that from 6 to 12 months old, there was no change in dendritic length in their branches. And this is the analysis showing for both the males on the left and females on the right, the top row for those cells that are in the ganglion cell layer, and those cells that are displaced. So there is no changes over 6 to 12 months old. This is our preliminary data that we looked at using the double transgenic model. So this is not the 3xTG, it has 1 less mutation. And this is a vertical section through the retina showing you a non-transgenic control at 2 years old. You can see the photoreceptors, the second layer of neurons show the bipolar and amacrine cells, or the inner retinal neurons, and then the single cell layer of the retinal ganglion cells. We look at one of this double transgenic AD mouse models at 10 months old, and you see some degeneration already occurring, but grossly normal. But by 12 months, so you have this rapid degeneration in all the cell layers, noticeably in all of the ganglion cell, the inner retinal neurons, and the photoreceptors. This is another vertical section showing the bipolar cells degenerate as well. Things are quantitated here on the side. And so these are at 10 months old, 12 months old, right? At 10 months old, it looks grossly normal. When we look at a 10-month-old mouse for AD double mutant for the melanopsin cells, this is now, instead of a vertical section, we turn it and do an en face or flat mount, and you can see the dendritic field here in a control. And in the 10-month-old animal, these are essentially almost all gone. And in fact, there's a little bit of thinning because you can start to see the first nuclei of the internuclear layer showing up here. So just I wanted to mention again, we did most of our studies not in this model because these mice die very young and the degeneration occurs very fast and very rapidly. So switching back to the 3xTG model, we look at total retinal ganglion cells in the mice, and we can see that control males and females, we see a loss at the 12-month-old time point. And then if we look at the M1 ipRGCs in the ganglion cell layer, we see that the 6-month-old females or males have a fairly high number of them per millimeter squared, so density, and that by 12 months we see a loss of these cells. We're still analyzing all of the ages in between, but we have looked at some of the synaptic markers or indicators of synaptic function, and these are female control mice at 12 months old. This is a flat mount. You can see the soma here and the dendrites. This is looking a little lower down, and then looking at the actual dendrites of in high magnification, you can see these boutons, and these are thought to be synaptic connections or represent synaptic connections to cells in that dendritic layer. If you look at 4 and 6-month-old 3xTG mice, they look relatively normal. They're quantitated over here. But if you look at the 16-month-old, you see, this is an example, you see a loss of these boutons. And in fact, that loss shows up between 6 and 8 months old, suggesting that even before we see significant cell loss by 12 months old, we're seeing synaptic changes or reorganization and perhaps a loss of synaptic function as early as starting shortly after 6 months. So that's kind of the end of the presentation. I'm just going to summarize now with the ex vivo retinal markers. We know that ipRGCs are stable in wild-type mice between 6 and 12 months old. And again, this is an older animal, but it's not a really aged model. This would be equivalent to a somewhat early onset Alzheimer's patient's age. So we see retinal degeneration occurs in both the 3xTG and the APP double mutant mouse lines. The degeneration occurs later in the 3xTG model, and this is consistent with them being not as severely affected. We see that ipRGCs degenerate early in the Alzheimer's disease models, and that cell loss may be preceded by alterations in neuronal connectivity. So taken together with the behavioral and functional data, it seems that early detection of ipRGC-dependent retinal functional biomarkers may provide us with a non-invasive tool, and that we could potentially use this for clinical evaluation for onset and for control should patients end up on drugs that are deliverable earlier in the disease and for progression. So with that, I'd like to just acknowledge all of the people that have contributed to this, shown in bold. My research group here at the University of Houston College of Optometry, Shreya Mitra and William Perez, are doing a lot of the analyses on the PLR right now, and the optomotor Sachin Parikh was instrumental in doing all of these experiments at UCLA. Rajesh Goit and Zhao Yuwang assisted with it. All of the OCT and ERG were done with Steve Nusinowitz and Eduardo Araujo at UCLA. Of course, Luis Perez de Sevilla Muller is the PI on the grant that we had. Brandy Recio participated in doing a lot of these studies. The PLR is a collaboration through Randy Carden and the mice from Samarhatar. And these are undergraduates that did their undergraduate thesis on the project. So I wanted to acknowledge them, as well as many, many other undergraduates that helped along the way. And with that, I guess maybe I'll just stay on the summary slide, or better yet, I'll stay on a nice degeneration slide so that this can really hopefully leave you with an impression of what happens in the retina, and that it is a really good way to look at neurodegeneration in both animals and people. Thank you.
Thank you, Anna, very much. Very nice data and interesting stuff. So we already have, excuse me, we already have quite a few questions, and I suggest that we dive right in. The first question relates to your ERG measurements, and the question was if you have looked at pattern ERG at all, since you're interested in RGC function rather than outer retina function in the Alzheimer's model.
Yeah, so that is a good question. The pattern ERG, for those of you who aren't familiar with it, You're basically placing an electrode. It's like an electroencephalogram. So you place an electrode above the visual cortex and you give flashes, and you can measure a couple of response waves to see if there's any loss in it. We have not looked at that because the pattern ERG is really good for measuring the retinal ganglion cell contributions, but not so good at detecting the ipRGC contributions. They're a small part of it. They project to other areas as well. So we actually have looked at the pattern ERG in the ipRGC ablated mouse line, and we weren't able to get very much. We weren't able to see many effects with that. So we did not look at that. That would also, you're also getting cortical degeneration or central CNS that's not the retina degeneration can contribute to that, right? Through if you see a loss in the LGN. And then in the visual cortex as well. So we have not, but that is definitely something that could be looked at and would again be like a good sort of global measure.
There actually was, yeah, here it is. There was a question about this. So in the case of Alzheimer's and using the readouts that you use, how can you differentiate between retinal contribution to defects that you see versus central changes in central processing?
Well, that is always the question that has to be asked. When you're talking about a behavior, I think it's a little harder to do. If you're talking about ERGs, it's obviously very simple to do. We just got the question about pattern ERGs versus a regular ERG. But if you're talking about behavior, that is, I think, something that studies like the ones that we're doing and many other people will look at the onset of degeneration or the onset of dysfunction, misplacement, synaptic connectivity loss in the brain versus in the retina. If we know that one occurs histologically earlier than the other, then I think that's the only way that we could overlay and say that this is indeed a function of the retinal neurons as opposed to a function of some decline in the and like the central region, the brain, whatever you want to. It's you know, you can't say CNS because that's the retina as well, but wherever you're projecting to. So I think that is a difficult thing to do. I can tell you one of the things that we didn't touch on here is that there are vascular markers for Alzheimer's disease as well, and this is being pursued in human imaging as well. So this is something where you would actually physically look at vascular changes and could be potentially able to determine that it's a retinal function, but indicative of some pathology going on in the brain as well.
All right, then there's a question about your light aversion experiments, and mainly whether, since mice are nocturnal animals, if it would not make sense for them to avoid light, so that would be a normal behavior rather than a sign of pathology.
Yeah, so this is a question that I do get a lot because people are used to. So this came up because very early on we started studying photophobia or neural circuits for light aversion or photoallodynia, and this is clinically defined as pain to light. But all of my friends were from behavioral neuroscience, so they would ask me, well, why are they afraid of the light? And the answer to why are they afraid of the light is because mice innately, even though they've been in the laboratory now for 100 years, they understand that they are lunch in this world for most predators. So just because they're nocturnal doesn't mean that they're innately light aversive, but in fact, if you habituate them to the chamber, it now becomes like home to them and they know it's safe. And I showed you in the one picture that even at 2000 lux, which is massively bright, mice still come out to the open. So they're not, as a nocturnal animal, they're not innately aversive, but they are very much afraid of any new place and any place where they could be exposed to a predator. Once they know a predator is not there, they're usually okay with the environment and the light.
Okay. Then a question about the melanopsin cells, the ipRGCs. Can you, from the way you look at it, can you really tell that they are degenerating, or could it also be that melanopsin expression is just downregulated?
Absolutely, it could be. So that's a question that we have for ourselves as well. So how do you determine if a cell is gone versus if a protein marker is gone? So we do those functional tests. As you saw, the results were not as clean cut as if you just get rid of all of the melanopsin cells genetically. So that is not a decisive measure either. So the only way that you can do that really is to find another marker that is specific for those cell types. And we're fortunate enough that in the retina, ipRGCs, melanopsin is like 99% co-localized with another protein called PACAP. So we're going to get some of those antibodies and take a look at whether or not the cells are gone, or if it's just melanopsin expression is downregulated, because both of those would actually have major implications for function. Because for one, you would still have the neural circuit that leads to, say, the suprachiasmatic nucleus for all of the circadian effects. That would still be intact, and you could have rod and cone input, but you wouldn't have the melanopsin flavor added to that. And it would also be indicative of a disease or a disease state of pathology in the retina.
Mm-hmm. Okay, and you showed this one graph that shows that the melanopsin-containing cells can have rather broad projection pattern into the brain, and not just how people often think going to the suprachiasmatic nucleus. Does that make the interpretation of the results more complex, or how does that play into the interpreting of the experiments?
I sure think it makes it a lot more complex. It's nothing like a neuron's going to a single site, but that's not how our brains work. So, I mean, the key is to, so you have to kind of map things. So for some of these behaviors, they've been mapped. There's a lot of great research in this field, a number of them. I'm not even going to say their names because I'll forget some of them, but that have done a great job of mapping where these projections are and then correlating loss of subclasses of these to specific functions. But it's still, you have a finer hammer to hit these. So it doesn't make it more complex. I think this would be like a great area in general for AI to sort of start combing through how these things to model how these things might work. But that's not what I'm doing just yet. So hopefully someone smarter than me out there will start thinking about how to model these at a grander level than what we've been doing.
Then a question, do you see amyloid deposition in the retina or just in the brain?
So definitely amyloid deposits are in the retina of both of these models, I believe. And this is work done out of Maya Coronio-Hamoui's lab at Cedars-Sinai in UC in Los Angeles. And she published a couple of years ago both antibody-based staining of human and animal model amyloid plaque or amyloid deposits in the retina. And she also used I forget whether it was humans or mice, but she used curcumin to look at in vivo amyloid deposits. These detection methods are tricky. The antibodies are not great. So it can be tricky to get it to work. Curcumin also has some trickiness to it. And people are now starting to look at hyperspectral imaging in vivo retinas, and this is able to basically give you a spectral scan breakdown of deposits. So there would be a characteristic deposit, for example, for a Lewy body or versus an amyloid plaque or other deposits. So they're starting to look at ways to do this type of imaging as a differential diagnostic in vivo.
But would there be a difference in the amyloid deposition, whether or not you have melanopsin knocked out, or is that completely unrelated?
It should be unrelated.
Mm-hmm. Okay.
I think it's not so much. It's more that whether it be the amyloid deposit, the vascular pathologies, the energetic pathologies, or the neuroinflammatory pathologies that are ongoing, and those are the four major aspects of Alzheimer's disease that are thought to lead to the neurodegeneration. Regardless of which of those it is, those are probably damaging the neurons, and that's why but there are going to be some that are more susceptible. Maybe there's more, and Dr. Hamoui's shown that there's deposits in the inner retina, in the ganglion cell layer, so it's not specifically just at the ipRGCs, but they might be more sensitive to the damage caused by it. They might be more sensitive to vascular or neuroinflammatory processes. We just don't know why they would be affected.
Okay. We have time for one more question. There are more questions still here, and we will post the answer. I just tell the audience we'll post the answers to this question in writing on our Journal Club website. I mean, we have to give Anna a couple of days to do the written answers, but we have time for one more question. From your work, knowing about the pupillary light reflex, what is your insight regarding predicting cognitive impairment in humans if one would use that readout of pupillary light reflex? How predictive is it of cognitive decline?
I mean, I would just have to completely guess. I know, I can tell you that in Alzheimer's patients, they've seen PLR deficits, and the specific set they've seen are, so let me just see if I can just quickly go to PLR. Right, so the deficits that they've seen are in a increased latency, right? So a slower response to constriction, a decreased amplitude, and a faster release or return to baseline. To me, those all scream ipRGC that the ipRGCs are still functioning in this, and that it's the rod or cone photoreceptors that are deficient. So I don't know that that there's been a good analysis of the onset of when these changes occur. And that's something I think that, again, we can sort of look at in mice to see not necessarily an absolute timing, but a relative timing to other phenotypes.
Okay. Thank you very much. We also received a lot of comments to thank you, and that it was a great talk from the audience. So I have to agree. I would like to thank the audience for many great questions and for your attendance and participation. And I hope that we'll see all of you in our next Striatech Journal Club, which will be sometime at the end of the summer. Thank you again, Anna.
Thank you for letting me speak here. And thank you for attending.
It was a pleasure. Okay.
Have a good day.
Bye-bye, everybody.